Hello everyone. Welcome to our Maxwell webinar. This is super exciting because it has been quite some time since our last webinar. And today we have a very special speaker who also published his paper recently within this year. Let's dive ahead to our program. For today, we will start with an introduction by me about Maxwell Biosystems and also about our new accent tracking assay. And then we will have our main presentation by Daniel Sommer from Obien University. So he'll be talking about impaired activity in motor neurons modeling ALS. And after that, we will have the Q&A for all the questions that you have. So anytime, please feel free to write in your questions. So if you are not familiar with this platform, you have some buttons, one for chatting with everyone. So you can just click chat. You can also see some messages from Maxwell Biosystems here about some papers or some links or some input or some just instructions on how to do things within Zoom. And then we have the other button, which is Q&A. And here you can add in your specific questions. And at the end of the presentation, we can just go through them and then have your questions being answered by Daniel or by myself. Okay. So is everyone ready? Everyone okay? All right. So let's begin. So welcome. We are Maxwell Biosystems, and our company's goal is to be able to provide electrophysiological platforms that allow you to be able to record activity from your cells. And this allows everyone to be able to advance scientific discovery and also to accelerate drug discovery for everyone. And the technology has been developed since more than 20 years at ETH Zurich with Andreas Hillermann's lab. And then in 2016, we found a company. Right now, we have 40 employees and we are growing. 50% are women with 17 nationalities. And we are here based in Zurich. We are broadcasting live. And if you'd like to visit us, just drop in a note and we are happy to welcome you here in our offices and labs. All right. So what about our system? So if this is the first time that you're attending our webinar, this is about our high-density microelectrode array. And we have this wonderful technology with 26,400 electrodes within a 2 by 4 millimeter square area. And the center-to-center distance between these two electrodes is 17.5 micrometers, which then allow you to be able to record activity of single neurons as well as those that are propagating along the axons. And that is also thanks to the very low noise readouts that we have. Additionally, any of these 26,400 electrodes can be used for electrical stimulation. So you can really manipulate every single cell that you're interested in your sample. Now, how do we record from these MEAs? We have two platforms. The first one is the Maxwell system, very handy, very cute. It's the size of my hand. And this is something. Okay, so, all right, going back again. So this is the Maxwell system. It's very flexible, and this you can use the single well HMEA Maxwell chips. And on the other side here, we have our Max2, our multi-well HMEA system that allows you to then be able to record all 6 wells at the same time. So we have a 6-well format and a 24-well format. And the interesting thing about this is that it has a built-in incubator, so you can maintain the environment for the cells quite similar to how it is inside your main incubator. And so these are our two systems, and depending on your application, you will find a solution that is right for you. So what are the things that you can do using this technology? So focusing on the neurons, we have 3 different assays. So the first one is the activity scan assay, wherein you're able to scan through all of the 26,000 electrodes to be able to identify the location of those that are active, the cells that are active. And here you can figure out, for example, the amplitude of the signal and also the frequency of the signal. In this functional image below, we are able to see the firing rate of the activity that is being recorded from the cells on top of the chip. And you can see that it really nicely shows you where the cells are. So here you see kind of a round shape, which is also reflected in the functional image. So imagine label-free, without any dyes, without any manipulation to the cells, you are able to follow the image, the functional image of these cells across multiple days throughout development. Next, once you know where your cells are, we can then focus the analysis and the recording to those active cells. And then we move on to our network assay. So here we have the top, let's say 1,000 cells or 1,000 locations where your cells are that are then being recorded at the same time. So we are able to identify whether there is some synchrony occurring within the network, represented by these network bursts. And in this case, you are able to then characterize whether your network is now matured or some developmental phenotypes that can be found either in like healthy cells or you're in disease models. And I think this is something that Daniel will also show to you later in his results. And finally, something that's really very exciting for us because this is unique to our technology is the axon tracking assay. And here you can identify the following. First, some sort of a morphology of your axons based on the propagating action potential along these branches, and to be able to identify the conduction velocity along every branch of these axons. So let me show you this video, which is just showing you our new axon tracking assay. So of course, these neurons are communicating with each other through these axons, transmitting the signal accordingly. And what the assay does is to be able to track this activity, really low signals, but really for each individual cells. So imagine you have hundreds to thousands of these in a multi-well plate that you can record all at the same time. And this is all applicable for Max 1 and Max 2. And thanks to everything automatic, everything that is done within the software in MaxLab Live. Okay, so just a few examples of applications where you can use our systems. Of course, 2D cultures, whether that is primary neurons or iPSC-derived neurons, you can plate them on the MEAs and get some results. You can also attach PDMS structures and be able to design some small structures yourself and identify their signals accordingly. Also apply some stimulation, and you can also, of course, apply these for brain organoids and spheroids as shown here on the right side. So these are just a few things about our HDMEAs and about Maxwell Biosystems. So as I mentioned, we have a growing team. You can see everyone here in these pictures. If you're interested to join us, please send in your application to hr@mxwbio.com, because maybe you are the next one here in this picture. So so whether you're in neuroscience, engineering, you're an entrepreneur, you're a computer scientist, you love stem cells, neuroscience, etc., you're welcome here in the company. So yes, so that's it. Now let's move on to our main presentation by Daniel Sommer. So Daniel is an MD student at Obien University in AG Catanese in the Institute of Anatomy and Cell Biology, directed by Professor Dr. Tobias Bockers. So for today, he will talk about his work that is also published in Frontiers in Molecular Neuroscience. So please welcome Daniel Sommer. Hello, Daniel.
Hi, and thanks for the introduction as well as the invitation.
Yes, our pleasure. So now I'm going to stop sharing and you can start sharing your screen.
Exactly. So, as already mentioned, I'm giving the main presentation today, which will be dealing with transcriptional and activity impairments in human ALS motor neurons. First of all, we're going to have some words about ALS. So, amyotrophic lateral sclerosis, which is a very heterogeneous late onset neurodegenerative disease, which mainly impairs the motor system and then leads very characteristically to flaccid and spastic paralysis in, of course, different areas. As I already mentioned, this is a very heterogeneous disease, which is also reflected by the underlying genetic causes. So mutations in more than 30 different genes are known to be causative for sporadic or familial ALS. So the sporadic variant is about 90% of the cases, familial ALS about 10%. And the most common mutation is a C9orf72 hexanucleotide repeat expansion. So the chromosome 9 open reading frame 72 gene, and then there's a G4C2 hexanucleotide in the intron 1. And if there's an expansion of this repeat, then this leads to ALS. And this accounts for about 34% of the familial ALS cases and about 5% of the sporadic ones. And this makes it the most common genetic background of ALS. Not only the genetic background is very heterogeneous, but also the clinical features. So partly due to the background, but also within certain genotypes, there is a huge variety of different disease progression types. And something that is very common in the clinical features, apart from the motor dysfunction, is cognitive and behavioral impairment. And 14% of the cases actually meet the criteria, so the diagnostic criteria for frontotemporal dementia. That is because of the genetic overlap. So there's some kind of an overlap between the genetic backgrounds of ALS and FTD. So far, therapy-wise, there have been 2 drugs, or there are 2 drugs available, which are riluzole and edaravone. Both of them just have an insufficient effect. We're talking about like months of increased survival in the patients. Some background information about the project that I'm going to talk about. So we are a synaptic lab. We are mainly focusing on the synaptic aspects of diseases, and especially neurodegenerative diseases in our group. And things that we have already observed in the past are like loss of synapses, increased size of postsynaptic protein clusters, and also what we could show was a downregulation in synaptic genes on mRNA level. So we have established some alterations in synapses in ALS, but there's also lots of literature. I just put some examples that we have observed ourselves here. And when we're talking about neuronal activity, there in iPS-derived motor neurons, there is a theory suggested, which is basically in the beginning of cell culture, there's an hyperactivity, and later on this hyperactivity decreases and switches to hypoactivity. So I've listed some papers here. Some of them described like early hyperactivity, other ones then later hypoactivity. And some of them, tried to give it or make this time-dependent context. For ourselves, we could also confirm the theory with on a biochemical level. We had in a previous publication looked at CRAB activation in ALS motor neurons over time. And here we could see, so CRAB is an activity-dependent transcription factor, so we used it as a surrogate parameter for electrophysiological activity. But we could see that there are increased phospho-CRAB, so activated CRAB levels in young ALS motor neurons, and then there's a decline over time, and at later time points, it's actually, in a way underactive. So the next question that we asked is, since there is a lot of discussion about this theory of early hyperactivity and later hypoactivity, we wanted to characterize the activity phenotype and later on have a look at what's its relevance for neurodegeneration. And since I already said, this is the activity most likely develops in a time-dependent manner, the first question that we had to ask ourselves was which ones are the right time points, so which time points should we choose for analysis? And therefore, we first of all had to answer the rather philosophical question of what defines a mature motor neuron. And therefore, we asked one of our collaborators to analyze or reanalyze some of his data that he already published previously. And we looked at the expression of synaptic genes in different cell types. So if we look at the PCA, it starts like with induced pluripotent stem cells. And from stem cells, we go by like motor neurons derived from iPSC over spinal cord homogenate, and then here is like singular laser dissected motor neurons. And we had a look at the, as I mentioned before, at the synaptic transcriptome and how synaptic genes develop during this maturation process. And we could see that there's a cluster of 35 genes that continuously gain expression during this developmental process. So having established that that one can use synapses or the synaptic transcriptome and therefore synaptic development to characterize the maturity status of human motor neurons, we went back to our own model. And what we looked at was in healthy cells, we had a look at the synaptic development. From our previous work, we already knew that at diff42, so days in vitro 42, which is like 6 weeks of culture, that up to this point in our ALS cultures, the number of motor neurons stays about the same. So there's no detectable neuronal loss going on until that point. So we used this as a reference point. And then we took an earlier time point. We chose diff21, so 3 weeks of culture. And we had a look into whether we have a synaptic development, so a gain of maturity between those 2 time points. And if we look at, so we did this by immunostaining, and we could observe that the presynaptic marker as well as the postsynaptic marker gains, also Bassoon and HOMO1 gain intensity over this, over this time, over this 3 weeks of more maturation, and also the colocalization is increasing. So what we have established here is that we have 2 different time points, and there's definitely a maturation process going on between those 2 time points. As I mentioned already earlier, we already know that there's no real neuronal loss until diff42. So just to confirm this, and also to confirm diff42 as kind of like the beginning point of neurodegeneration in our neuronal cultures, we looked at LDH leakage. LDH leakage is a stress parameter. And for the early time point, we could not detect any levels of elevated stress in our ALS cultures. Whereas at the later time point, the LDH leakage starts to increase, and therefore, this gives us like an idea that cellular stress also is increasing. So the next question that we ask ourselves, how about the neuronal activity between those 2 time points and what happens here? And that's why I'm here today, because here the Maxwell company gets into the game. We were looking at this by one of their products with the Max1 system. And what we could see was that we indeed have an initial hyperactivity in our ALS motor neuron cultures. So if we just have a look at the left side here, this is again, so this is always the same 2 cultures. So this is a healthy culture at diff21 and the same culture at diff42. And here it's an ALS culture at diff21 and the same culture at diff42. And the first thing that we can that is very impressive here is that at diff21 we have this hyperactivity in the ALS cell line, which is not that much evident anymore at the later time point. So if we look at the quantification, then we can see this pretty strong early hyperactivity, and then there's a drop in activity, and at diff42 the mutant cells are, from their activity point of view, are a little more similar to the controls. Another point that we had a look at was also the network behavior or the network properties. And we could see that in the beginning it was a little bit inconclusive at diff21 because there was some inter-individual differences between our 2 cell lines. But at diff42, we could see a decrease in the network burst frequency So there was less of these events where a lot of cells fire simultaneously, and this gives us we can conclude some kind of synaptic imbalance at a later time point. Okay, so having now looked at the neuronal activity, the next question that came into our mind was, what are the underlying cellular alterations? And we opted for a transcriptomic analysis. So we did a transcriptome of each of the genotypes for each respective time point. And then we compared the two genotypes at each time point, and then also longitudinally within the genotypes. So first of all, I'm going to talk about the differences between the two genotypes at each time point. We're starting at the earlier time point, and the most important thing about the early time point is that we could see an upregulation of synaptic transcripts in the mutant motor neurons. So we could see this upregulation in neuronal activity, and along with that, we could see a upregulation in synaptic processes. So for example, here GO revealed like chemical synapse transmission, transsynaptic signaling. And also with gene set analysis, gene set enrichment analysis, we could see like synaptic membrane and neurotransmitter uptake. Before we now switch to the later time point, I want to quickly highlight those two numbers. So we could see that there was a downregulation of 426 DGs and an upregulation of roughly 280. And now if we switch to the later time point, you can see that these numbers actually decrease. And this is interesting. So apparently from the early to the late time point, both genotypes get more similar. And this is also reflected by the fact that by geobiological processes, we could not detect any upregulated process. So earlier we had this upregulation of synaptic terms. Now this is not evident anymore. Gene set enrichment analysis already gave us some idea that there might be more apoptosis in the mutant ones. And also this term with RNG-TBase binding is kind of like interesting because C9orf72, the gene product, is involved in that pathway. In the next step, as I already mentioned before, we did longitudinal analysis in the C9orf72 mutant motor neurons. And what we could or detect here was, of course, what we expect if we detect or if we analyze the transcriptome during a maturation process. So there was a downregulation in like this developmental processes and also in cell cycle, stuff like this, since neuronal maturation goes on, and then logically these processes get downregulated. On the upregulated side, we could detect, in fact, increased stress, which again confirms what we could already see by LDH leakage, and also autophagy. It's known to be a common feature in ALS that there's some dysregulation in protein degradation. So this also is in line with what's already published. Something I want to highlight here is that also neurotransmitter transport popped up as a process that's downregulated. And therefore, we wanted to go a little bit deeper into what happens on the synaptic level between those two time points. And therefore, we just had a look at the synaptic DGs between diff42 and diff21. So we used SynGO to filter those and analyze those. And what we could see was that there is down and upregulated synaptic terms in both of the genotypes. But there's a pretty big cluster of downregulated synaptic differentially expressed genes associated with chemical synaptic transmission, transsynaptic signaling. So apparently there is some downregulation in terms, especially annotated to neurotransmitter vesicle release. So we have this decrease in synaptic terms. I just want to, since maybe this was a bit confusing over the last few slides, I want to have a quick interim summary just to describe the phenotype again. One might ask why I put this PCA here now, because I put it here because I think it's a very nice visual way to show what we detected so far, at least for the neuronal activity and synaptic transcripts. So first thing is we can see a very clear separation between diff21 and diff42, which is, for those of you familiar with PCA, is very nice in terms of internal validation. But so what we can see here, that there is a separation at the early time point between healthy controls and the C9orf cells. And upon time, actually this becomes less evident. So the separation between healthy and ALS is less evident. And that's because at diff21, we have this increased neuronal activity and the increased synaptic transcripts without any levels of elevated levels of stress. And upon neuronal and synaptic maturation at diff42, we have this increased levels of stress, as we could show by LDH leakage, as well as the transcriptome. But we have this drop of neuronal activity and the loss of synaptic transcripts in the mutant cells, and also this reduction in synchronized firing. So having now described this phenotype, we were asking what to do about it, and is there any beneficial effects of activity modulation? And we chose apamine for this, to modulate the activity. Apamine is a potassium channel blocker that blocks SK channels and increases the firing rate by lowering the after hyperpolarization after each action potential. And apamine has already been shown to be neuroprotective on the one hand side by our group in IPS-based models, but also completely independently at another group in a Drosophila model. And the first thing that we wanted to do, just as a proof of principle, was to show that apamine, or that our cells respond in the way that we expect them to do so on the electrophysiological level. And that's again what we did with the Max one. What we could see after the application of apamine, there was an increase in the firing rate, which is, here you can see the quantification, and here you can see some representative network activity plots. So we can elevate the firing rate and also make the cells fire more synchronously. So as you might remember from earlier in my presentation, there was, apart from this reduction in firing rate, there was also, compared to controls, a lower burst frequency at the later time point. And both of these parameters are actually increased by the application of apamine. And therefore our cells are, as we could show here, still do react in the way that they expected to, or that we expect them to do. And in the next step, we wanted to have a look at what are the further effects of apamine. And therefore, we again looked at, on the one hand side, at the LDH leakage, where we could show that apamine actually decreases cellular stress at this later time point. And also we had a look again at the synaptic transcripts by PCR, and we could see that just a 3 days treatment of apamine is already sufficient to at least restore, or to at least increase the expression of some synaptic transcripts in our motor neuron cultures. So some of these transcripts are the ones that were formerly found to be downregulated, and some of these are actually, they originate from the first figure that I've shown. So from this cluster of 35 synaptic genes that increase upon maturation. Yes. So what conclusions can we draw from the findings that I've shown you today? So C9orf72 mutant HIPSC-derived motor neurons display an early hyperactivity, at least in our in vitro cultures. I put here also this citation that, yeah, it came out pretty much at the same time as my paper. And they found something similar. So they also found they found an early hyperexcitability, which might be an underlying cause of the hyperactivity that we found. But again, or still, there's a lot of questions that are risen by this fact. So some people just suggested it might be just a cell culture artifact. So in terms of answering that question, one would like go to an in vivo model, look at what findings are there in animal models. But there is also a huge discussion about early hyper or hypoactivity. The theory that we support here is that it depends on the motor neuron subtype. So some of you might know that there is like different types of motor neurons in terms of alpha, beta, gamma, but also in the alpha motor neurons, there are certain subtypes which can be characterized according to their electrophysiological behavior by patch clamp. So maybe there are more vulnerable subtypes, or there are more vulnerable subtypes, and they might, or their electrophysiological phenotype might differ from other subtypes. Another question that has to be risen here is, what's the role of neuronal environment? Apparently also the neuronal surrounding in the cell culture plays a role on this activity phenotype. And the last question that remains is, what's the clinical correlate? Since there is not a real clinical correlate found for this early hyperactivity. So still the clinical and biological relevance, or not relevance, but impact of this early hyperactivity is to be discussed. Another conclusion that we can definitely draw is that there is an age-dependent loss of electrophysiological activity, and this is accompanied by a decrease in expression of synaptic transcripts. And the last thing that is of note is that the restoration of neuronal activity, as we did with the upamine experiments in the old C9orf72 mutant cultures, has beneficial effects on neuronal activity, but further also on synaptic transcripts and cell stress. And this confirms that the loss of neuronal activity that we could observe from diff21 to diff42 is actually relevant for neuronal survival, since if we restore the activity, the survival gets better. Okay, so then I'm actually at the end of my presentation. I would like to thank some people, which is especially Professor Böckers, who's head of our institute. And special thanks also go to my supervisor, Alberto Catanese. And also thanks to everyone else from our group and our institute that helped with this project. Also huge thanks to all of the external ones, so our collaborator Richie Ho from CEDAR Sinai, but also our university internal collaborators that helped with this paper. Yes, so okay.
All right. Thank you so much, Daniel. Really, really nice presentation. It's very nice that you have shown, the correlation between synaptic information and how that correlates to neuronal network activity as well. So very nice. Thank you so much. Before we start with the questions, please again, information for all, you can type in your questions here in the Q&A button right in the tool, and just write them down, and then we're going to answer them within this presentation. And just also quickly, we would like to run a poll. So I will launch a poll now. All right, so the poll is ongoing. Let me scroll through the questions now. Oh, Daniel, we have quite some questions. Are you ready?
Yes, I am.
Yeah. Okay, good. Okay, thank you so much, everyone, for answering the polls. All right. Good. So next, we would like to then go through the questions. First, from Heike Blokus. Hello, Heike. Very nice to have you here. So Heike said, great presentation. She said on slide 9, does she interpret it correctly that there is no increase in activity in the healthy line from day 21 to day 42, although you have seen an increase in synaptic or maturation markers?
So we did not detect a significant increase in neither firing rate nor network burst frequency. That's correct, yes.
Okay. So but have you seen kind of this increase like earlier time points, maybe like before day 21?
We didn't do network scans before div 21. So we had a look at so we did activity scans starting from div 14, but the results are similar to what I've shown here.
All right.
So we couldn't really detect a difference here.
Thanks a lot.
But I read a paper actually. They were also looking into these aspects, and they also did not really find until a similar time point at the 42 that the neuronal maturation is actually that much or has that much progressed that there is really a lot of synaptic activity. And therefore this in the context, yeah.
Okay. So it's matching with what you have seen as
exactly,
well. Yeah. Okay. So next we have from David Biasniata. Hello, David. So he asked, does the reduction of the mean firing rate go together with the fraction of active electrodes? In other words, do neurons fire less frequently, or did you end up with less neurons when the mean firing rate are reduced? Did you analyze this?
I did not analyze it in detail, or I did not analyze it statistically. I have to put it this way, but of course I looked into this. It is true that there is a decline in active electrodes from the early to the later time points. That's true. But so on the one hand side, first, I don't think that, so this is a phenomenon that actually occurs in control and ALS motor neurons. And if we look at the firing rate or the mean firing rate between the two time points, we can see that the mean firing rate in the control cells actually stays rather stable around, something like 0.5 hertz. That's on the one hand side, and on the other hand side, still, since I mentioned earlier that there is no real, or we could not detect neuronal loss by immunostaining between those two time points, there should be still the same number of cells within our cultures. So I don't think that this is like a phenomenon of a smaller network or something like this.
Okay. Thank you. The next question we have is from Pravina Managaran. Hello, Pravina. So she said, fantastic presentation. So Pravina was wondering if you were able to show a correlation between your MEA results and other methods, such as transcriptomics analysis, like synaptic transcripts, neural activity-related DEGs, etc.
We did not do a statistical correlation, but I think, as I have already presented here, like what do you call it? In terms of sense, or in terms of what we observed, this was like there was like a similar development in both of for the different methods of analysis.
All right. Okay. Thank you. All right. Next, we have from David again. So David asked, could you tell from your markers what proportion of inhibitory neurons you have, and if SK is mostly expressed expressed in that subpopulation? So in your
Yes,
protocol,
yes,
yeah.
yes. So we know from earlier works that most of our most of the cells in our cultures, more than 90%, are motor neurons.
That's high.
Yes. So the subset of neurons in our cultures is more than, or 90%, or a little bit more than 90% motor neurons. I don't remember, maybe Alberto can quickly step into if we did, but I don't think that we specifically looked at inhibitory neurons.
Okay. Well,
Um,
and we can then follow up with David after the webinar.
yeah, okay.
Okay. Thanks, David. Next question from Kevin Marinus. Hello, Kevin. Very clear presentation. Thank you so much, Kevin. So what kind of interneurons does your differentiation protocol yield? So did you have interneurons as well?
Still, yeah, there is a short, or there's a small amount of interneurons in our cultures, but by heart, I don't remember the exact phenotypes.
Then let's follow up.
But the question aims at a similar thing as the previous one.
Yeah. Okay, so well, the next question is about stimulation, but I'm not sure if you did electrical stimulation. So maybe
Only pharmacologic stimulation.
Yeah. Okay, so maybe we skip that question for now. Next question: have you found UNC13A reduced in ALS?
That's something that I would have to look at the tables.
All right.
But also the tables of differentially expressed genes, they are a supplementary material on the paper. So if you just click the link, you can also access the data.
Yep, that's correct. So you can just look through the chat and you'll find the link to the paper there again. Yes. Okay, moving on. We have so many questions for you. Next, all right. So nice work. This hyperactivity was seen in neural outgrowth, meaning higher growth in diseased lines.
So for this paper, we did not analyze stuff like dendrite length or something like this. So I can't answer this question.
Okay.
Or I don't know. This might definitely be something that might be interesting. Definitely. But we haven't analyzed that, no.
Okay. How does your outlook look like? Like from what you have seen so far, how does it, like what are your future plans? Do you plan to kind of characterize it more or proceed to new cell types?
So there is some things that would be cool to look into in the future. So on the one hand side, we just did all of this for the C9orf 72 mutant cells, but I mean, as I stated earlier, there's like a lot of different ALS genes, and it would be interesting whether this phenotype holds true for more of the different genetic backgrounds, or there is some indications that it is, that it holds true for some others, but to what extent and to what subgroup this is actually relevant for. And what's also very interesting, or in our opinion also important, is maybe to, we found apamine to be neuroprotective in our cell cultures. There was it's neuroprotective in Drosophila models. So the next step here would be to develop a substance that works similar or has similar effects on the neurons and could be used in patients.
Yeah, thank you. So we have some questions related to apamine actually. So one is from Fraser McCready. Fraser said, great talk. Did you try treating the control networks, the wild type ones, with the apamine as well? And if so, did you see more oscillatory pattern of the activity in these control networks?
We did not treat control cells with apamine, since in our opinion, this was a not so much relevant question, since our idea or our what we wanted to do was to improve the phenotype of the ALS cells.
Yeah.
And therefore we just tried it in the ALS cells.
All right, okay. We have one more here from Shan. Hi, Shan. Since apamine could rescue the phenotype, did you also see transcriptomic change in your RNA-seq data in terms of potassium channel genes?
This maybe if Alberto can answer this, but so there is, or we already earlier published a paper where we used apamine and also did a transcriptomic analysis before and after, or with the apamine treatment. That's the paper from Alberto from last year.
Yeah.
I think it's answered there.
Okay. Then we'll get back to you, Shan, from Alberto's paper.
Yeah.
Okay. We still have more. I think we'll have just 2 more, and then we can answer the rest of the questions by email. Is that okay? Okay. So the next one is from Navdani. Hello. Thank you for the presentation. Is the level of LDH related to the neurodegeneration, or we can see it's increased even if we have very confluent culture?
Okay, so the experiment we did here was we analyzed it differently or separately for diff21 and diff42 and always just compared it to controls. So maybe there is a general rise in LDH levels over the course of time. This might be true. But for us, it's more relevant the level, in comparison to controls.
Yeah.
So I would say it's yes, we can trace this back to neurodegeneration since it does not occur in the healthy cell lines. And if there is some problem, or not a problem, but if the confluency rises, this would also happen in the controls.
Okay. All right. So our last question from an anonymous attendee. Did you try to prevent the hyperactivity as early treatment to prevent the loss of activity later? So I think you applied the apamine around the later days. If you applied it earlier, would that kind of alter the activity eventually?
That is a question. We did not do this, but there's a paper from already 2014 by Brian Weinger or his group. And they were the first ones to describe this earlier hyperactivity. And they also developed a pharmacological treatment, retigabine, that actually decreases this initial hyperactivity. And what they found was that the neuronal survival gets better at this very early time point if you reduce the early hyperactivity. There has been so they did a follow-up study in patients and tried retigabine in patients. But so far it is not known to produce clinical improvements. So they could detect like in electrophysiological measurements on patients. They could see that there is some effect of retigabine, but there was no clinical effect, at least to what's published by now.
Okay, interesting. All right.
Yeah, that was actually also kind of a motivation to do this paper, that maybe this whole early thing is just too early, and the neuronal maturation going on is like is responsible for some artifacts that might lead to results of questionable relevance.
Yeah. Okay. Thank you so much. So Daniel, is it okay if I share my screen?
Yes. Oh, sorry.
And if we were not able to answer your questions, because whether maybe I missed them in the chat, or I was just not able to go through them because of the time, we will get back to you via email. So we will transfer these questions to Daniel and Alberto, and then I will get back to you via email. So thank you so much, everyone. And please feel free to contact us at info@mxwbio.com, or schedule a call with one of our scientists using this Calendly link here. And you can find us in the upcoming conferences right below. So thank you so much, everyone, and wish you a nice day. Hope to see you again in our next webinar. Bye for now. Bye. Thank you. Thank you, Daniel.